want to show it to you. This is a skate. So I mentioned that we don't see those guys too often here. This is actually one that was collected by a collector for the St. Augustine Aquarium, and they mistakenly identified it as a butterfly, right? And tried to maintain it. It died in captivity. It probably died because these guys don't really do very well in captivity anyway. And this is it's known as an oscillated skate. So this is it's also known as a Texas skate sometimes, and you can always tell by these patterns here. But you can see some of the unique characteristics. Again, we've got the disc here. This is the cartilaginous rostrum, and then you see the pectoral fins kind of move both rear and to the forward, and then connect to the rostrum. And you've got the eyes on the top here. The spiracles right here. See that there? That there. So you can see how, particularly on this side, how that lifts up. And we've got essentially a naked body here, but there's thorns here. You might be able to see those guys there, as well as this row here. See how they're raised? This is when I mentioned the presence of what I referred to as, and we'll talk about them during reproduction, but malar and alar thorns are basically thorns that occur within the skate, particularly in the males, and they'll usually be here as well as up here. And the reason for that is the skates kind of lay on top of each other. The male's usually on the bottom and rotates the clasper forward to inseminate the female. And so by having those thorns on there, it kind of allows the female to kind of catch a little bit and keeps them together during mating. And but then in both the males and the females, you would see this midline of thorns, and in some cases even a greater number of them here on the tail that are in some cases pretty sharp, pretty raised. And then you've got your two caudal fins as well. And then of course he's male, so you've got claspers. And here's the gills on the underside. And you can see the claspers are actually quite large with regards to the length of the body. And you can always tell the maturity of a male. This is, of course, pectoral, and then here's your pelvic fins. Remember I mentioned that the skates like to punt, so they're like using their pelvic fin like little legs. Well, these are the little portions that are used like little feet. They're called the crus. And so these guys move back and forth and allow this guy to kind of move along the seafloor. The claspers grow significantly during the maturity period. So these probably only started off like this, and then they grew considerably. You can tell maturity on the basis of clasper calcification. And so this here is heavily calcified. It's but then if you look at the ends of the clasper too, you can tell maturity based on their ability to flare. And so this is the flaring here, and you can probably see that little cartilaginous spike in the center. In addition to growing longer, what happens is it grows these little cartilaginous rods that are almost like little spikes that allow them to adhere to the female reproductive tract and kind of embed into it. And so again, this rotates forward and then opens up, and essentially you see the little tube there. This is essentially a groove, so that the in this little region here, the reproductive tract would empty semen into here, and it would slide down there. You can see this little knob here. You can see it on the side too. I'll be talking more about these later, but in when we go over the reproductive cycle. But in the skates and the stingrays, they have these little organs called the clasper glands. That's what this is. And it's a gland that secretes this very thick, white, viscous fluid that we're actually not too clear about what it does, but it gums up as soon as it hits seawater. So the idea might be that it reduces the dilution of the semen as it's being passed to the female. Some suggestions were that it might produce a copulatory plug, so that when male mates with a female, it's gumming up the works, essentially. And the other suggestion is that it's very lipid rich, and it might provide nutrition to the sperm in order to maintain survival until fertilization occurs. And again, here's the little teeth, and I don't know how well you can see, but they're cusped teeth. They are. This is mostly a fish eater, so you can hear that they are kind of pointy. And again, all the innards are very compacted. This is the you can get a good view of the again the pectoral fins here, but you can see the pelvic girdle in these guys. So this is a cartilaginous rod right here, and then see how it rotates forward and then back. And so all of the organs are basically in here. The heart is right here. And again, here are the gill slits here. Questions? Okay, so let's take a look at some of the sharks. So I've got two little black tips that I pulled out. I thought that I pulled out a shark nose, but they're both black tips. And we've got both a male and a female. And so again, if we look back at the pelvic fin, here's the male, and you can see how small these claspers are, demonstrating the immaturity. You can see how they just kind of flip. They have no calcification to them. They're just soft and spongy. In the case of the female again, sorry, it's been cut here, but you can see that there's no clasper that's extending from the pelvic fin. With the black tips, identification is pretty easy because you have a black tip on the fins, but you'll note that that is on the pectoral fins. Black. You've got a little bit on the dorsal margin. It kind of gets a little bit more prominent with the older animals. It's very small here. Here's the second dorsal. It's a nice black tip. Same thing with that. Whoop, just cut off. And then you'll see it, of course, on the particularly on the lower margin or the lower lobe of the caudal fin. But note that there is no black tip on the anal fin here. And same thing here. This is a diagnostic characteristic that allows you to differentiate between the black tip and the spinner shark. The spinner shark also has black tips, and ironically, the spinner has a black tip on the anal fin, but the black tip doesn't. Black tip was named before the spinner, which is funny. And spinner shark is named for its ability to jump out of the water and perform these really cool little spins. So we see a lot of the things that we talked about already. Of course, here's your 5 gill slits, eyes present. Again, you've got this pattern of ampullae around the mouth, particularly around the mouth and around the rostrum. Here are the nares, here and here. Of course, you've got the mouth. You don't see really any visible signs of the spiracle in this guy. These guys are generally active swimmers, and the spiracle is much reduced in this group. You do see some of the pores here alongside of the body, and some of those are linked to the lateral line system. And again, here's the rostrum. So we've got our first dorsal, missing second dorsal, caudal fin, heterocircle caudal fin. Okay. Upper lobes larger than the small lobe. Anal fin, pelvic and pectoral. Same thing like that. And then when we kind of cut this open, again we've got those muscular bundles I call myotomes. Right? So the muscle is in these very specific compartments. It has that kind of V shape to it that you'll see in other chordates as well. The largest thing that's visible when you open up the animal is the liver, of course. And again, the liver is playing a role in maintaining neutral buoyancy because it is very lipid rich. And you'll see differences in the size of the liver and the amount of lipid within the liver depending on the ecology of the species. You're going to see greater amounts of lipid. Obviously, something that swims more actively than something that would be benthic. In addition, the liver is playing a huge role in detoxification, and then also in breakdown of lipids in the diet. And one of the things that generally visible, it's not too visible right here, is right about here. This little brownish thing, usually it looks kind of greenish, but this is the gallbladder here. And so the gallbladder is producing bile that aids in the breakdown of lipids, and that's being discharged into the intestine a little further down through the bile duct. So if we follow the mouth, and this is actually showing you, this was a cut through the compartment here. This is the pectoral girdle right here that I'm pointing at. So we cut the pectoral girdle line up, and you can see the heart. The cavity that we have the heart in is called the pericardial cavity. Normally that would be separated from this cavity, which is the alimentary cavity, also known as the peritoneal cavity. So if we move the liver to the side, and we've got one, and there's two big lobes, and there's a little center lobe. We've got a whole bunch of connective tissue here that holds the remainder of the organs together. And the other really prominent thing is the digestive tract. So that's what I'm holding here. And basically this here is stomach. And if we follow it forward, you can see how this gets a little beige here. And that's showing the transition to the esophagus. And so, of course, food's going down the mouth. The esophagus has got, the lining of the esophagus has got a little finger-like villi. And the idea is that that's aiding in moving food downward. But then when you get to the inside of the stomach, when you get down to the stomach, you don't see any of the villi anymore, you see folds. And in the case of the folds, the role of the stomach is to digest food. And so the idea is that you're increasing the surface area for the production of digestive enzymes in order to break down food. The stomach moves forward, so we're following stomach down here. We get to what's referred to as the pyloric sphincter, which is going to control the movement of food from the stomach into the intestine. And so this right here, this is the anterior-most portion of the intestine. This is this thin strip right here that I'm pointing at. You see that there's this other little tissue here that's attached to it, but see how it's separated? So this guy here is the spleen. Spleen is an immune organ, and it produces white blood cells, produces red blood cells as well. Cartilaginous, they don't have bone marrow, and so most of the blood cell production is occurring within the spleen. So food's moving down here to the stomach. It is movement is isolated, or not isolated, but halted by the sphincter, which allows stuff to go in at a controlled rate. And this anterior portion of the intestine is the duodenum, and the duodenum is where the bile ducts as well as the pancreatic duct empty into the gastrointestinal system. And so in the case of the bile duct, again, the bile duct's producing bile, and the gallbladder's producing bile, it's putting it into the gallbladder. And the liver's producing bile, putting it into the gallbladder. That's being discharged to the bile duct into the intestine, and what it does is it acts as an emulsifier. It breaks up fats in order to increase the surface area for enzymes to break them down. When you follow that duodenum down, and you get to this area where it kind of increases in size. This is the ileum, but this is also what we have always referred to as the spiral valve or scroll valve. You always hear about the spiral valve, and it's kind of like it's like a little staircase, right, in the intestine. And the intestine is so much shorter than our small intestine, but like the stomach folds, that the rugae allow for a greater amount of surface area for digestion. The spiral valve produces a greater amount of surface area for absorption. And so that's where things like monosaccharides, amino acids are being absorbed, and they'll be absorbed to a greater extent because of all the folds that occur within the intestine. When I said scroll valve, what I mean is that in some species of chondrichthyans, the intestine is this sort of staircase scroll, but in other cases it's called a spiral valve. But in some cases it's just a what we refer to as a scroll valve, which is that the membrane inside is basically just folded on top of each other. So when you cut it open, it looks like a whole bunch of papers that are just like all falling out. But again, the whole purpose is to increase surface area. And then if you follow that out, that is intestine out to colon, and then out to the rectum and the cloaca. The cloaca is the common opening here, which receives discharge from the digestive tract, reproductive tract as well. One other organ that is usually visible, and you can see it here. So this little guy here. This is the rectal gland. And the rectal gland's role is it's a salt secreting organ. And so as we'll talk about in the sharks with regards to osmoregulation, the sharks are osmoconformers and ionoregulators. What it means is basically that they increase the amount of salts within their body so that there's limited osmosis, there's limited movement of water between themselves and the environment. So they're conforming osmotically. But they ionoregulate because what's happening is that the amount of sodium and chlorine specifically within their body is a third of what's in the environment. And they're eating all of this seafood. And so they tend to concentrate sodium and chlorine. Rectal gland secretes sodium and chlorine into a lumen, and then that's discharged through the digestive tract and removed out of the cloaca. The other thing that I'll highlight here is if you look at the mesenchyme, you can see that there's see this guy here. This is the pancreas, and it's broken up into multiple lobes, and you can usually only see it within this connective tissue. Mentioned that the gallbladder releases bile that's involved in lipid metabolism. The pancreas of the sharks is similar to our pancreas, so there's an endocrine and an exocrine component to it. The endocrine component produces insulin and glucagon for sugar metabolism, just like us. In the case of the exocrine component, that's producing enzymes to break down carbohydrates, to break down fats. But in addition to that, it's also producing buffering solutions. And those buffering solutions are discharged into the duodenum so that when you have all of this acidic material in the stomach moving into the intestine, the buffering solution buffers it before it gets into the intestine, so it maintains the stability of that intestinal wall, which needs to be at an alkaline pH. Now, what I've done, and you'll see this, and a couple of things you won't be able to see too well. I'll show you some other images of it once we get to reproduction. The gonads are very small within these individuals because they're immature. They're on the body wall here, and they're really not easy to make out. And this line here that I'm running over